Okay, I hope we are all refreshed after the break and ready for the next module. As I said, module 1, we practiced from plain text file, tabular file to a data frame, and that data frame is almost ready for statistics or machine learning. The second module is more for genomics, because I know lots of audience have a strong interest in genomics. And then in genomics, there are like so many formats. People give you some file, and it's our job to turn that file into some matrix or data frame so we can do statistics or machine learning. And the second module is more focused on the genomic data. How do we manipulate genomic data and visualize genomic data? Okay, so the first one is again by Dr. Se-Young Ko. Se-Young has done a lot of development in our group about writing Julia package for parsing the genetic data, and that's the content of his lecture. Yeah, so it's here. Yeah.
Hello, it's Se-Young Ko again, and for this section, let's talk about easy manipulation of genetic variant data in Julia, and specifically about the single nucleotide polymorphisms or SNPs, because they are most widely used for genetic variant analysis. A SNP corresponds to a nucleotide position on the genome where some degree of variation has been observed in a population, with each individual have one of several possible alleles at that position on each pair of chromosomes. So the alleles are often classified as a reference allele, the allele on the reference genome, and the others, which are called alternative alleles. The most widely used repository for common SNP information is dbSNP, where each SNP is indexed by an identifier beginning with RS, short for RefSeq, and this is often referred to as RefSeq ID or RSID. And we commonly use biallelic SNPs for many analyses, so we will assume biallelic SNPs for this tutorial. The genotype of each sample or for each variant is commonly coded in numeric values of 0, 1, and 2, 0 for homozygous reference allele, 1 for heterozygous reference and alternative, and 2 is for homozygous alternative allele. And sometimes when genotypes has uncertainties, it is represented in dosage after imputation. Given the posterior probabilities of each genotype, dosage is computed as weighted mean of the 0, 1, and 2 numeric values weighted by the posterior probabilities of each genotype. So they have values between 0 and 2. Often the data formats for genetic variants include information of M samples and N SNPs, and an M by N table or matrix containing observed allelic type at N positions for M individuals. A common type of analysis for this data is GWAS, genome-wide association studies, often testing a statistical hypothesis variant by variant. Significance of each SNP is assessed by some type of regression, where the trait to test is on Y, and for covariates, we have SNP, which is genotype or dosage, and we adjust with additional covariates such as age, sex, principal components, and others. In this workshop, we learn 4 widely used file types for genetic variants and how to manipulate them in Julia. We focus on accessing genotype information variant by variant, as this is common workflow for a GWAS-based applications. We learn also how to compute some simple properties of SNPs, such as minor allele frequencies. Let's begin with why there are so many formats. Genetics, like many other scientific fields, is a fast-moving subject driven by academics. So in the early stage, they devise formats that best suit their needs rather than using what is out there. And as the field matures, it converges to a couple of dominant formats. The formats covered in this workshop are considered to be a relatively dominant ones, and there are many, many more out there. So we will talk about 4 different formats and 4 packages to read in these data in Julia. Let's begin with the variant call format, or VCF. This is a text-based format, which is de facto standard for variant call information. The text-based format is the most intuitive to represent genetic variants, and it is the most flexible to include diverse information on the samples and variants. And with raw text, not compressed in any way, it is highly flexible, easy to parse and interpret, but the file size may be prohibitively large. And with the scale of recent genetic data, with dataset like UK Biobank having near 1 million subjects and millions of variants, the storage needed for storing a raw VCF file is prohibitively huge. And a common approach to remedy this is storing the data in a compressed form, such as GZ format, as we see in module 1, and decompress it at analysis time as a stream. However, there is a trade-off between stored file size and time needed for decompression. So with the compressed format, the pros would be it will be reasonable file size. However, cons is that it takes too long to decompress. Here's the basic usage in Julia with VCF2.jl, which is mainly written by Dr. Benjamin Chu, sitting here. We'll load the VCF2 package, and with the open VCF function, we open the file and we print out first 35 line of our example data named test VCF.vcf.gz. So here's what text VCF format looks. The first lines of the file starts with hash sign. They are all header lines, including version of VCF and what information are in there, and some formats for genotypes, dosage, and genotype likelihood, and additional information there. Here's the reference genome, and the last line of header is column names: chromosome, position, ID, reference value, accuracy value, quality score, and others, and the sample names. Then the main body starts with each line has the information of each variant. So here is chromosome number for the first one, position, ID, and and for each sample we have genotype, dosage, and genotype likelihood of each of the 3 possible genotypes. And here's the second line beginning with chromosome number and so on. And in VCF2's package, to access the number of records and number of samples, we have the n records sample, n records function. And for number of samples, we have n samples function. So we have 1,356 variants and 191 samples in this file. And information on samples and variants can be retrieved using the variant call format package, which VCF2 package internally uses. With this cell, we can get 191 sample names of this file from the header. Then information of each variant is accessible by this cell. It reads in first 30 lines of records and then prints out chromosome position IDs, which can be multiple. So it is in the list form, and reference value and alternative values where multiple alternative values are allowed. So it is again in the list form.
The VCF reader function, that's offered by the VCF close package or the variant call format?
This is from variant call format.
Is that package still being active variant call format?
At least it is alive. Yeah. It was at least alive when Ben was with us. Each row of a VCF file represents a single variant. So it is natural to parse the genotypes or dosages variant by variant. Inside the VCF tools package, we have the function copygt to compute genotypes for each, to parse genotypes of each variant, and copyds function to parse dosages of each variant. So using VCF tools package and the reader from variant call format.jl, we pre-allocate the vector for marker data to reduce the memory allocation for efficiency, and we also pre-allocate the place for sample IDs and record names, position, and IDs and others. So for the and we read in first 30 genotypes using the copygt function. The first argument is the space we allocated for the marker data, and we input this reader, and we have we have other keyword arguments. And after reading it, we do some statistical analysis, and just for illustration, we will show the mean level of the genotype values by doing just mean G. This model keyword argument shows which model to use, and for our 0, 1, 2 encoding, it is additive and imputes the missing values with the mean of the genotype levels.
Some means are 0 means those are mono allelic. Is that what you mean?
That is right. Everything is 0 in those variants.
But it's just a small sample,
Yeah.
small section of your whole famous.
Yeah, it is just 191 people. Small section of a huge pool of people, I guess. So that is how to do for VCF format. Then the second format is Blink-1's bad format. And to remedy the issues with text-based format, to be able to rapidly process the variant information while keeping the file size small, various binary file types has been devised. And for a well-celebrated large-scale GWAS2 Blink-1, it internally has a bed format, which is basically a compact array of 2-bit representation of genotypes. We have only 3 or 4 possible cases, so it is reasonable to use just 2 bits for each entry. And this is suitable for high-performance applications directly dealing with genotype matrices, such as iterative heart thresholding and admixture analysis for estimating ancestry. However, its major weakness is that it cannot contain it cannot contain imputation information when there is uncertainty in genotypes. So all we have is these four values: 0 for monoallelic allele 1, homozygous allele 1, and 1 is for missing, 2 is for heterozygous, and 3 is for homozygous allele 2. Advantage is small size, and it is fixed with format suitable for massively accelerated computation, such as GPU acceleration. And it is best to parse n variants of a single sample. However, its disadvantage is that it only contains these four values, and it needs external files for variant, which is .bim format, and sample information, which is in .fem format. Those two formats are text-based. And another disadvantage is that reference value, alternative value designation is not supported, and this has caused a lot of confusion between researchers. In our meta group, we have the package named SnipArrays.jl, and we read in bed format data like this. Information of the ith sample is accessible by this code, because we have person information field for SNP data. And to access jth variant information, we have SNP info field, so we can get SNP information like this. And for genotype of sample I and variant J, it is accessible by this line. We have an array interface for this one, and the value is 3 in hexadecimal representation, and this is encoding based on this table, so it means value of 2 in 0, 1, 2 encoding. Genotypes of each variant in numeric form can be accessed by pre-allocating a vector for genotypes and then using the copy2 function to this array. And SNP array has to be accessed in this view macro. And for Plink file, what is listed as second allele is encoded. So you might notice difference between this one and this one. If it is desired to run analysis based on the alternative value count, the values have to be reversed, which means subtracted from 2.0. And if we subtract this from 2.0, the values will be same as in VCF result. This has led to a lot of confusion, and some later projects began to put the reference value first, most notably UK Biobank. Before that, it was common to put reference value as the second value. The third format is Oxford's Pgn format. This is native to Oxford statistical genetics tools such as impute2 and SNP test, and it employs variant by variant compression scheme, which is pretty well tailored for GWAS applications. And because it is Britain-based format, the UK Biobank impute data is distributed in this format. Compression algorithm used are gzip and z standard, which is backed by Facebook or Meta. Its advantage is very small file size, and if we only include genotype information, further compression for the fixed width genotype representation is possible. So it is smaller than the bed format, and it admits variable precision. So each entry can be 1 bit, 2 bit, 3 bits, 4 bits, 8 bits, 16 bits, and up to 32 bits. And it allows dosage and phase information to be included inside the format. However, it needs an external index for random access to get to like 16th variant. It is hard to do without external index file. Without external index file, it is hard to get to like the 16th variant. And compression is not tailored for genetic context. We are just using the compression algorithm that are there. So it is very slow to access genotypes of a single individual. Because to do so, we need to decompress the entire Pgn file. And another disadvantage is that reference value, alternative value designation is not supported. The package for this is pgn.jl under Alphamento. I wrote this package. And with the Pgn structure, we load this example data file, test Pgn. The sample names are accessible by the samples function. And since how to iterate across the variants in Pgn files depend on whether we have index file or not, we support an indicator interface for variants. If you're familiar with Python, it is Julia equivalent of generator defined using yield statement in Python. This iterator is accessible by the function iterator. So we will iterate over the variants using this iterator function, and we will parse for first 30 variants and get variant information. We have access to functions such as RSID, chromosome positions, and number of values, and list of values. And we are assuming biallelic situations, so we always have 2 alleles and length to list for alleles. And in this file, first one is the alternative value and second one is reference value. Dosage of each variant is accessible by using the first value dosage function. And because first value is the alternative value of this situation, we get this result, same as what we got for VCF files. First value dosage function requires the Pgn object and variant object from this iterator. Now let's get to the fourth format, which is under development by Plink team. Because the previous Plink format is very efficient but very limited to genotype information, they wanted to include dosage information and phase information here. And some cutting edge GIS tools are not already incorporating this format. Its advantage is that it has very small file size, similar to Pgn. And again, it is variant by variant compression. And it allows dosage and phase information. And the compression is based on patch list. So it saves the difference from some reference, difference from all 0 vector or difference from all 1 vector, or considering linkage disequilibrium, which means that the variance nearby has high correlation between them. it can sometimes save the difference list from the previous variant. And doing so makes it 4 times faster than reading in Pgn file with similar precision. Its cons is that it is still under development, and it is difficult to write a parser by admitting too many forms, including the whole bed format. And it still requires external variant and sample text files. Here is how to read in Pgn format using the package Pgn files, which I wrote. And usage is pretty similar to Pgn. We have Pgn structure, and we can print out number of variants using n variance function, number of samples using n samples function. And we also have the iterator interface. And in order to get genotype values, we use get genotypes function. This G, the first output of get genotypes function, is a genotype vector, and it has values 0, 1, 2, and 3. And Plinka has actually changed their encoding for version 2. So it is 0 for homozygous reference value, 1 for heterozygous, and 2 for homozygous alternative value, and 3 for missing. And to avoid reallocations for iterative genotype expression, we can pre-allocate G and reuse it using get genotypes exclamation point function. We often put exclamation point to indicate that the values are mutated in place in Julia. So this is how we get mean level of genotypes for the first 30 variants in this example file. Similarly, alternative value dosages are available through the function named L-value dosage. And because in order to decipher dosage level, it requires genotype information. So space for genotypes and space for dosages are both required for the L-value dosage in place function. So we pre-allocate both dosage levels and genotypes here, and run the iterator to get the dosage levels. Information of each sample and variant is available by reading in psem and pvar files as a data frame. It is not yet supported by pgnfiles.jl. And pvar file notably admits regular VCF files as pvar file. So this is how we read in sample information from the psem file. And this is how we read variant information from the pvar file. And all the lines starting with hash sign is a header, and the last header line being the column names. And we know that is 26th line, so we put this here, and then we get information on the variants. And we have reference value and alternative value designation in this file. Exercise I propose would be computing binary frequency, which is widely used for determining if a variant is rare or not, and in GWAS it is used for measuring information content of a variant. One of the measures that computes information would be the ratio of actual numerical variance of dosages or genotypes and expected variance from the binomial model. So by modifying the cells like this, so putting some code here, you can compute minor allele frequency and information contents. So the exercise is to compute binary frequencies and determine the major and minor allele and the variance ratio measure. And you can print out those for each variant in each line. And transformation between the file formats can easily be done using Plink package, Plink program. And Pgn bed and Pgn files are transformed from the original PCF file for this tutorial. And these are the commands I used for this one. Here's the concluding remarks. While we focused on variant by variant access of the genetic variant data, many of the packages we have seen has various utility functions, such as filtering out the variants with low minor frequency or low genotype success rate, or filter by chromosome or merge between files. And in case of the fixed with bed format, Plink1, we support high performance linear algebra on the genotype matrix, which supports multi-threading and GPU computation. Under our Open Mendel group, we have various packages for processing genetic variant files. For 4 GWAS applications, we have ordinal GWAS.jl by Dr. Christopher German. It is GWAS for ordered categorical traits such as disease status. And another is trajectory GWAS.jl, GWAS for continuous longitudinal phenotypes using a modified linear mixed effects model. And it tests both mean level effects and within subject variability effects. And for the bed format, we have high performance applications using fixed width form. Mendel HT.jl, it rather than using GWAS with variant by variant testing, it uses penalized regression model. And with opennet mixture.jl, it estimates as ancestry of samples. It is Julia re-implementation of highly celebrated admixture software in C++, and it is 8 times faster in multi-thread setting and 35 times faster using GPU. And other packages we have are variance component models, Mendel Impute, which for phasing inference and genotype reputation, and trait simulation for quickly simulating phenotypes under a variety of genetic architecture, and quasi-copula.jl by Dr. Sarah G here, analysis of correlated data with specified marginals with a flexible quasi-copula distribution. That's it for genetic variants for formats. Thank you.
Thank you, Seyong. I think here we can defer the questions to the end of the module. Looking at this example, I'm still amazed. I mean, a high-level language like Julia can do this kind of operation. I believe if we want to write some, as statistical geneticists, if we want to do something like this, essentially, if you don't use Julia, your choice is C or C++. You have to dig into those low-level language to do this kind of operation. And I'm still amazed you can do this using a high-level language. Okay, so now I think you are convinced it's pretty easy to ingest genetic data into numbers, so you can do number crunching. So next lecture is how to visualize genetic data using Julia. And it's my great pleasure to introduce Dr. Ming-Soo Kim. So actually Ming-Soo is a real doctor, because just in the morning, just in the morning, Ming-Su was doing surgery here at the UCLA hospital. I'm kidding. Yeah, were you? I mean, I know you are doing rotation. So Ming-Su is a MD, PhD student. I see Ming-Su just finished the PhD part like 1 month or 2 months ago. And currently Ming-Su is doing a rotation in surgery department here at the UCLA hospital. I have to say, not many data scientists can do surgeries, right? so Ming-Su, go ahead with a genetic Maki package Ming-Su has developed.
Hello, everyone. My name is Min-Su. I'm a medical student here at UCLA. Yeah, up until a month ago, I was a PhD student in the human genetics program, also here at UCLA. Today, I'll talk about a Julia package that can be used to visualize genetic and genomic data at scale. It's called Genetics Maki. So I think we can start off by discussing why you should try using Maki. Maki is one of many Julia plotting packages that are out there that are open source. And there are two main reasons that I can think of. The first is that if you use Maki, plotting millions of data points becomes quite easy. So shown here is one example. So here I'm showing the data from this region on chromosome 6, 24 megabase to 36 megabase. It's a fairly large region. This region is also known as the MHC region. It stands for Major Histocompatibility Complex Region. And MHC region, interestingly enough, is the single most complex region in the human genome. And this is because there are lots of immune-related and brain-expressed genes that reside within this region that are important for normal human physiology. As a result, over the course of human evolution, a lot of mutations and polymorphisms have accumulated in this region. And weirdly enough, not much genetic recombination is occurring within this region. As a result, many SNPs within this region are extensively correlated with one another over an extensive genomic range. So if you look at an LD reference panel that I used to generate this plot, there were 66,000 SNPs. And shown here is a heat map of correlation among these 66,000 SNPs. So this heat map corresponds to plotting approximately 2 billion unique data points, which is quite impressive. If you try to draw the same plot in other computing environments, such as ggplot in R, your computer basically will crash. It cannot handle the heat. So if you're interested in plotting billions or millions of data points with ease, I highly recommend trying out Maki.
Mentioned that it's kind of washed out on our screen, but if you look at it, the version that you can access directly on your computer, you'll see in more detail than you're seeing on this projection.
Yes, that is correct. And the large square here corresponds to class 1 region, MHC class 1. The second large square here corresponds to class 2 region. The small square here corresponds to class 3. And these squares are extended region. The remaining squares are the flanking regions. And I should have put a legend here, but I forgot to. But anyway, so if you're interested in human genetics, the genes that reside within this region come up over and over again in human genetics studies. So I would highly recommend also doing some additional reading, for example, this paper. So the second major advantage of using Maki is that it comes with an advanced exceptional layout tools that allows you to visualize figures that are very complex. So this is one example figure that I've generated for my upcoming paper. And so this is a raw output from Maki and Genetics Maki. So without any further modifications, such as in PowerPoint or Adobe Illustrator. So this figure is, of publication caliber, and I'm quite happy with where it stands. And it's actually quite complex, given, as you can see here, the spines or the borders of the plots don't really align perfectly, either vertically or horizontally. So Maki's default layout tools allows you to draw these complex looking figures, and it also allows you to customize and place either legends or color bars in somewhat of an arbitrary position. So this is also pretty powerful compared to other, for example, ggplot2 extensions that are widely used out there. If these examples look interesting to you, I'll highly recommend checking out the official documentation of Maki. the documentation is mostly driven by examples. It should be quite straightforward to follow. And if you're interested in gaining finer control of the individual components of of a plot, I'll also recommend taking a look at the API page in the Maki's official documentation.
So all these plots are made by your package.
Yeah, so yeah, just to clarify, this figure here, I mean, you don't have to know what the meanings of these figures right now, but this figure here, the panel A, panel C and D, and this rightmost figure is made by the package that I wrote, Genetics Maki. The panel B here is drawn by using the default bar plot that's made available for from Maki package. So yeah, and all these individual plots are put together using the default layout tools of Maki. So everything here is raw, as it's an output as it is, without any further modifications. And yeah, this is quite impressive, at least in my perspective. And
so you actually use this part in your paper.
Yeah, exactly. Yeah. And yeah, another thing to note here is that reproducible science also involves being able to reproduce all the scientific figures in its entirety. And Maki, by using Maki or Genetics Maki, you can easily attain that goal. And oftentimes, a lot of bioinformaticists or human geneticists spend quite a lot of time trying to draw these individual plots and manually combine them in, for example, PowerPoint or Illustrator, and that can be very frustrating and time consuming. So Maki allows you to avoid such issues. So I'll definitely highly recommend trying it out. Yeah, even if you're a heavy ggplot2 user. Yeah, so then we can talk about Genetics Maki, this package. So the goal of this package is to facilitate visualization and interpretation of genetic association results. And this is achieved by visualizing hundreds of genetic and genomic data simultaneously in a multivariate fashion. So we're currently living in a biobank scale era where we have access to genetic association results for hundreds of phenotypes for 20 million variants, SNPs. So it's become very important and essential to be able to visualize these results somewhat simultaneously in a multivariate manner, to not only for our sanity check, but also to gain novel biological insights. So as I said before, Genetics Maki builds on top of Maki. So obviously it's supposed to work with Maki, and it also under the hood, it's also utilizes other Julia packages such as SNP arrays that Seyon just mentioned, and also data frames package for tidying and munging data. Yeah, so this is another example plot that's generated by using Genetics Maki. So here we're looking at the screen 2A locus. Locus is synonymous with genomic region, in case you guys are not aware. So yeah, so we're looking at this region on chromosome 16, around 2.5 megabase window around green 2A. And green 2A is an antisense gene, meaning 5' end is at the downstream and the 3' end is upstream. And green 2A gene is quite interesting because it's a high confidence schizophrenia risk gene that's been prioritized from rare variant studies in whole exome sequencing. So yes, so if you look at GWAS results for schizophrenia, lo and behold, it also seems to harbor a fairly strong GWAS signal in the 3' end. So there's some convergence of common variant and rare variant signals here. And so you can try to look at the green 2A association results, not only for schizophrenia, but for many other phenotypes. So here shown are the results, association patterns for 40 or 50 phenotypes. You can see that bipolar disorder right here seems to harbor a similar or the same GWAS signal compared to schizophrenia. And this kind of makes sense in light of biology, that they are the bipolar disorder and schizophrenia tend to be comorbid with one another. So there's that. But if you look at other phenotypes like intelligence or cognitive ability and educational attainment, there seem to be distinct GWAS signals that are in the 5' end of this green 2A gene. So presumably these GWAS, these genomically significant variants, play a different downstream biological or molecular role for this particular gene. And this kind of observation where distinct GWAS signals are associated with a spectrum of phenotypes is also known as allelic series, just for you to be aware. Oh yeah, so I forgot to explain this. So for phenotypes that are harboring genomically significant variants, GWAS signals, the most strongly associated SNP, also known as index or sentinel SNP, is highlighted in diamond, and their RSID, their SNP ID is also labeled, and the rest of the SNPs within this genomic region are colored with respect to their LD. So the stronger the red color, it means these variants are in higher correlation, higher LD with the sentinel or index SNP. Okay, so yeah, so we can come back to this really complicated MHC locus. So as I told you before, this locus harbors around 66,000 SNPs, and we can really take it to the next level, at the phenome scale or phenome wide. So here I'm showing you the MHC association pattern for 110 phenotypes. So here, basically, this is quite big data. So I'm showing you the association results for 6 million data points, more or less. But and plotting 6 million data points as a scatter plot, it's not that impressive. Many other plotting packages can handle it. But here I'm also calculating correlation LD for each phenotype, and yeah, so this is quite computation heavy, and only Genetics Maki or Julia can handle this. It will be a nightmare to draw a similar plot or the same plot in ggplot, for example.
How long could be all code?
Yeah, so yeah, this one is actually quite a large amount of computation. This requires a large amount of computation. This took 20 minutes on my personal laptop. So all the figures that I'll be sharing with you today as examples were drawn on my personal laptop. This model for my laptop is specified here, just the MacBook Pro. And yeah, so on my personal laptop, visualizing these 6 million data points with correlation calculated, it took around 20 minutes. Some people might say that that's a while, but keep in mind that this type of plot cannot be generated in any other platform. So yeah, so yeah, so just, yeah, I mean, this is not too important, but you can see some psychiatric disorders here, neurologic disorders here, neurobehavioral traits right here, some ophthalmologic conditions, disorders right here, some gynecologic conditions right here, autoimmune disorders, some GI disorders, some cardiovascular disorders, some dermatologic conditions. And you can also see on the right side, you can also see GWAS MHC association pattern for complete blood count, basic metabolic panel, and urine analysis. So these are some common blood tests or urine tests that physicians order for in or out patients. And yeah, so the striking observation here is that the MHC locus seems to harbor at least one GWAS signal basically for every phenotype, 100 phenotypes. So which means that this region is highly pleiotropic and have a very interesting biological, I guess, role. Yeah, so if these types of plots look intriguing to you, I would recommend checking out the documentation for Genetics Maki package. The documentation is also driven mostly by examples. In the documentation, I've also tried to share as many know-hows and tips that I've gained over after visualizing, drawing these types of plots more than 1,000 times. So hopefully they're helpful to those who are interested. Now I'd like to share and run some example code for visualizing this type of locus zoom plot. Due to the limited memory that we have, we're only going to visualize a single locus for a single phenotype or disease. So I'm actually currently in a general surgery rotation, and we see a lot of hernia cases. So I figured that we might as well take a look at a GWAS for hernia, just out of curiosity. And interestingly, just last month, a paper that has run GWAS on different types of hernias have been published in Nature Communications. So there are different types of hernias, such as inguinal hernia, umbilical hernia, ventral hernia. But of these, inguinal hernia is the most common one. So I decided to focus on this. And luckily, the authors of this paper has made their GWAS summary stats publicly available. So I've kind of pre-downloaded the results for you. And in order to draw a locus zoom plot, you also require gene annotation data, transcriptome annotation data. And for this, we can use GenCode version 39. Yeah, and you also need some kind of reference panel to calculate correlation or LD. And for that, we can use 1,000 Genomes Project reference panel, which is publicly available and as a result, most widely used. So yeah, so you can try to load the modules, necessary modules or packages in the usual way. So genetics Maki and Cairo Maki are going to be used for visualization, CSV for reading in different types of files, text-based files, and data frames for tidying or munging data, and SNP arrays for reading in the Plink files for the 1,000 genomes. So I like to draw my figures as if they're going to be published. So I like the font Arial. So I'm just going to use the function that's provided by Maki to set the fonts. And in this code chunk, I'm going to read in the gene annotation. So I'm using here CSV read function. I'm reading in the object as a data frame, and GTF file by default does not have a header. So I'm going to specify the column names for the 9 columns, and they are tab delimited. And the first 5 lines of GTF file are usually comments. So I'm going to skip those lines. Yeah. So if you're interested in genomics, it's also a very good idea to become familiar with GTF files, file formats, because they are very widely used for all sorts of purposes. And the last column, the info column, contains rich information, metadata about each gene and isoform. So as you can see here, the 9th column or the info column contains gene ID, and it's not showing here, but it also contains information such as transcript ID and other information. So we can try to parse this column by using a function that's provided by genetics Maki. It's called parse GTF. So we are going to parse this data frame. And then once this finishes running, you can see that in addition to the 9 columns that came in as a default, you now have gene ID column, gene name column, gene type column, and transcript ID and transcript support level. Okay. Yeah, so in order to reduce memory intake, I'm just gonna select the columns that are going to be used over and over again. So, and for reproducibility, I'm just going to make sure that the dimensions of this data frame is in line with what we would expect by using this assert macro. Yeah, so and then I'm going to read in the 1,000 genomes project reference panel, and I've pre-subsetted them to only the individuals of European ancestry. Yeah, so that was quite fast, and I'm also going to read in the GWAS summary stats because we need this for visualization. Yeah, so this is the GWAS summary stats for just the chromosome 15. Yeah, so this is tabular data. And one thing to note here is that a lot of the publicly available GWAS results come in a very heterogeneous format. So you can imagine they might have different column names, and some of the rows might be missing, or some of the rows might not be valid. They might be funky. So we have to harmonize them in a nice manner so that we have easier life trying to plot these results. So yeah, so
Where did you get this? You want
so this was I got it from this paper. So this paper shares a link to their GWAS summary sets in their data availability statement. So that's why I just downloaded. Yeah. Yeah, so in order to harmonize these summary sets that come in a variety of flavors, you can try to use a Lange sum stats function that's provided by genetics Maki. So once you run that, you now have column names that are harmonized. So SNP ID, chromosome, base pair, alleles, effect size, z-scores, and p-values. Then so you can imagine running the same function over and over again for hundreds of GWAS summary stats that are publicly available out there. So yeah, so once you have harmonized your results, then you can try to, figure out how many genomic significant loci you actually have. So, so you can try to run this function, find GWAS loci. So, unfortunately, for this phenotype inguinal hernia on chromosome 15, it only seems to harbor one genomic region that is genomically significant. So the index SNP for this locus is on chromosome 15 and on this base pair position, and it has this p-value. It just barely passes the usual genomically significant threshold, but it's still significant. And if you want to, and you can definitely apply a more lenient p-value threshold by using this keyword argument p. If you do that, there seems to be at least one more locus that is defined to be a genomically significant. Yeah, so once you identify the genomically significant loci or locus, you can try to find the closest genes within that locus or loci, and you can do that by using this function, find the closest gene. And in order to identify the closest genes, you need a gene annotation. That's why you pass in gen code. And so for this single locus, for this single index SNP right here, the closest gene was this gene called TUBAP4. And yeah, but you might be interested in finding the closest genes, finding the genes that are closest from the 5' end, not necessarily from the 3' end, because you might have strong prior belief that these GWAS variants tend to regulate promoter, tend to reside in the promoter region. So in that case, you can pass in this keyword argument called start. And even when we run this method for this function, we still get the same gene, which is reassuring. But one thing to note here is that this gene is a pseudogene, so it's not a protein coding gene. And in real practice, you might actually want to find protein coding genes that are closest to your index SNP. So in that case, you can pass in this keyword argument called protein coding. Try to run it. So in this case, this ADAM-TSL3 gene is the protein coding gene that's the closest. Okay, so what I like to do in practice is I like to do a lot of sanity checks. So what I do is I go to Google and try to look this gene up. And if you go in here, this gene is apparently associated with Ehlers-Danlos syndrome. Ehlers-Danlos syndrome is a very well-known Mendelian disorder that has defects in the connective tissue. So, inguinal hernia, obviously something might be going wrong in the soft tissue or connective tissue behind the skin. So I think this gene makes biological sense. So that's, yeah, so. Yeah, and finally, you can try to find the closest gene from the 5' end, and also that is protein coding. And once again, we get this particular gene ADAM-TSL3. Now, next, we're going to draw a locus zoom plot, and this is the function that you can call to draw a locus zoom plot. This kind of looks complicated, but it actually isn't not. It's just a bunch of Maki syntax. So the plot that we would like to draw is this one. So here, this plot has 3 layers, 3 rows. The first row is just a scatter plot of genetic association results without any coloring. The second plot is just the same scatter plot, but the SNPs are colored with respect to their LD or correlation with the sentinel or index SNP. And the third row is a gene track, and it shows all the protein coding and non-coding genes that reside within this region. Okay, so this is the figure that we want to get at, and yeah, I'll try to quickly explain what each line does. So we're going to pass in a list of genes. This could be a vector or this could be a tuple, and we're going to iterate for each gene in this list of genes. And we're going to define a genomic window of 1 megabase pairs. So we're going to visualize more or less two megabase pair around each gene. And by using this find gene function from genetics Maki, you can find the location of each gene. So we're going to determine the chromosome and the start and stop sites of each gene. And then by using this window definition, you're going to come up with the range, the genomic range that you're going to ultimately visualize. Okay. And then you can try to subset your GWAS results to that genomic region of interest. So this is what this code does. There are many ways of doing this, and this is just one syntax. You don't necessarily have to follow this. And then we're finally going to start plotting stuff. So for Maki, we always have to declare a figure first, and I like to always draw my figure as if they're going to be published. So by using this resolution keyword argument, you can set the size of your figure. So these are the units of points. So 792 corresponds to the height of a letter page, and 306 corresponds to half the width of a letter page. So that's what I want for my publication. And then once you define the figure that you're going to draw on, then you also have to define the axis where the actual plotting occurs. So since we have 3 rows, 3 plots, we define 3 axes. So this is like a list comprehension. This is a Julia syntax. And yeah, so in the first first row, the first axis, we're basically going to plot a straightforward scatter plot. This is the command. In the second figure, we're going to plot a scatter plot, and in addition, color snips with respect to their LD with the index snip. So you can supply an LD keyword argument here. And the rest is basically just Maki syntax to fine-tune the plots for publication quality. So for the first 2 rows, we're going to label them. We're looking at the same GWAS inguinal hernia that was published in 2022, just last month. We're going to set the row of each figure as 30 points. This is what I like. You can try different points if, yeah. And then in the third layer, axis 3, we're going to plot gene bodies, gene tracks, and this is the function that you can use. And this function returns a single output that gives you the optimal row size that should be used. So you can set the row size of this figure as such, and then we're going to label the last row with the appropriate genomic range. This is what this part does. And here we're going to add a color bar. I mean, you always need to add a legend or color bar to make your figure as clear as possible. And here I'm also going to add a y-axis. Yeah, and just for clarity, I'm also going to label. I'm also going to draw vertical lines that labels the gene start and stop sites. And I'm also going to plot horizontal lines that signify the GWAS threshold, the significance threshold. And you can control the row gap, the gap between the rows and the gap between the columns in this manner. You can try to get rid of unnecessary space by using this function. And you can save it. So yeah, so let's try to run this. So the initial run is going to require compilations. As a result, there's this issue of time to first plot that Dr. Zhou mentioned in previous session. So it takes some time in the first run, but after the first one, things should be running quite fast. Yeah.
How do you know to harmonize if we get USOMers that
My recommendation is to run the Munge sumstats function for all GWAS summary stats, even though you might be highly confident that this summary stat is very trustworthy and very well of very high quality. My recommendation is to just run it regardless, so that you have the same data format for all phenotypes. Can you say that?
One and two, could be one studies filming, could be one, could be A1, A2, 100.
Yeah, so what it does under the hood, what it does is it has a dictionary. So so A1, A2. So for example, for the allele 1, allele 2 example, what it does is it converts everything, all the keys in the dictionary to, for example, A1, A2. But I guess I'm not explaining this too eloquently, but
You say you manually
yeah, exactly. Yeah. So I've looked at like at least 200 publicly available GWAS summary sets out there. So I've manually curated. This is a lot of scut work, but yeah, I've manually created the most common column names, and I've created a dictionary so that we can harmonize the column names in a nice manner. That's right. If it cannot find it, it gives you an error message. So in that, then you can try to manually change your column name to something that's more reasonable. Yeah, so it finished running. As you can see here, 80% of its runtime was spent on compilation. So this is not good. So we're just gonna run it again once more. Yeah, so the second run finished in 15 seconds. It's not too bad. So I'm happy with where it is. So yeah, so this is the final output. Yeah, so this is the final output, updated output, and yeah, so here we've only visualized one locus for one phenotype. That's pretty easy. This can be done in R or ggplot, but ultimately we want to visualize these GWAS summary sets for hundreds of phenotypes across many different loci. So if you're visualizing 2 megabase window, then the human genome is 3 billion base pairs long. So in order to cover the entire human genome, you have to visualize around 1,500 loci for hundreds of phenotypes. That's quite a huge amount of computation. But genetics Maki allows you to draw these types of plots in a reasonable amount of time. But anyway, so how do you exactly draw these phenome scale local zoom plots? I don't have time to run the code for you in this session, but I'll just give you a general workflow. Let's say you have run a GWAS and would like to visualize genome-wide significant loci for your GWAS programmatically with hundreds of other GWAS results and relevant functional genomic annotations. How do you go about this? Well, you first, as I said before, try to munge GWAS summary sets using the munge sum sets function. And then you try to save each GWAS result as either an arrow or Parquet file. So loading these hundreds of GWAS summary sets, each of which is at least 2.3 gigabase large, that's infeasible. That's not possible. So to load them in a memory efficient manner on your personal laptop, I recommend converting them and saving them as arrow files. And then once you do that, you can find GWAS loci for your phenotypes of interest by using the find GWAS loci function, and you can iterate through these loci, subsetting the relevant genomic regions from the arrow or Parquet files that you had saved. And then you can add any other functional genomic data, such as gene tracks, as separate layers as needed. So I actually have an example code shared in another GitHub repo. So if you're interested in this type of visualization, yeah, just you can try to check that out. Yeah, so genetics Maki also provides a variety of many other functional plotting utilities. You can go through the documentation if you're interested in it. One of the plots that you can generate is the Manhattan plots. So here, so here I'm visualizing schizophrenia GWAS results across different years. So with increasing year, the sample sizes have dramatically increased, and as a result, the people have identified a lot more GWAS signals. So I'm visualizing all these Manhattan plots simultaneously using genetics Maki and Maki, obviously, and yeah. So yeah, this is cool. And this each row contains around 10 million SNPs. So you're visualizing around 40 million SNPs here. So this is going to cause some pain and problem in ggplot, but it can be easily plotted in Maki, genetics Maki in Julia. So yeah, so for anyone who's interested in a big data visualization and drawing figures with complex layouts, I would highly recommend trying Maki.
How long it takes to make this for?
Yeah, that's a good question. I don't exactly recall. I drew this plot like a while ago, but I think it was definitely less than like 10 minutes. Yeah, so yeah, so yeah, that's it. Thank you.
Thank you so much, Min-Soo, for the amazing talk. I always like talks with lots of fancy figures and plots, and this is certainly one of them. And even better, Min-Soo showed us how can you produce these plots by code. It's just amazing. By the way, also I want to mention, don't treat this workshop as totally like coding and so on. Make it also a social event. If I know people, we have different people with different career stage, from elementary school to postgraduate. If we are interested in MD, PhD, how does that program work? So you should, Min-Soo is the one, you can talk to him. Yep. So yeah, let me introduce the third talk of module 2 by Dr. Alfonso Landorus. So Alfonso is another computing guru in our group, very knowledgeable about high performance computing and so on. He obtained his PhD degree in biomathematics, I think 1 year ago, right? And currently he's a postdoc fellow working with Professor Ken. So let's welcome Alfonso. Yeah. Thanks. Include the fonts.
Can I get rid of the,
Oh, yeah.
yeah, where is that? It's on the
Oh,
view.
yeah. You close the
Yeah,
other
go ahead.
kernel. Sorry. Let me- Oops. 91. And Maki. Okay, yeah, okay. Yeah, thanks. Sure. Yeah. Okay, great.
Well, welcome everyone. I'm going to talk about, this isn't my package, but it's something that I found very useful in my own work, just working with data sets that I had nothing to do with in terms of creation. Essentially, the issue is that oftentimes you run into a problem of like, where do you store all these different data sets that I want to run through, say, some sort of analysis pipeline. So for my work, that analysis pipeline really means I'm benchmarking a bunch of stuff with my method, right? I want to push a bunch of different data sets through it. Where do I store all of these things? And more importantly, how do I make it so that other people can very easily access those data sets again to reproduce my results, so they can either, verify that my method works as advertised in a publication, or just so that they can go in and play with things. There might be a lot of settings that they can configure, and they want to be able to do that, right? So that being said, the real work for this is due to this Julia user, Ox in a Box, and his collaborators that came up with the data depths package, because they from my understanding, there are researchers that work extensively in machine learning, and so they have some methods and that they wanted to test there. And this other Julia user, Checksum, who created this basically interface to the UCI machine learning repository called UCI data. You can look at that package, but it's essentially a way to very easily access a lot of UCI data, UCI machine learning repository data sets in a standardized format. So if you work a lot with those data sets, this is a really nice package to use, because it uses data depths and basically will make your life a lot easier, as described in this short presentation. So we can scroll down here. The authors for data depths came up with, or rather published a paper describing their experience and kind of the motivation for why they developed this package. And essentially, it just boils down to this piece I highlighted here in the abstract, which is they wanted a way to handle different static data sets in a reproducible way. So here, static just means that as far as anyone is concerned, that data set is never going to be updated. There's no additional records. No one's going to go in and add additional columns or rows. It exists in this particular state, right? And here, that reproducibility might mean I may need to go in and access that data set from the server somewhere. I might have to manually create an account somewhere to log in to access that server. We just want a way to document the whole process of being able to obtain that data and run any sort of pre-processing pipeline so that we can readily integrate that data set into whatever workflow that we're trying to execute. And so they identify these three issues that come up when you're trying to work with these different data sets. One of them that I kind of mentioned in the beginning is storage location. I have my machine. I'm going to store things in my favorite folder location, but somebody else that's working with this data might be working in a completely different computing environment than you. They might be operating through some sort of cluster that they're accessing through a terminal, and they may not have very tight control of writable locations for themselves, right? So they may be working in a resource limited environment. They may have to go in and compress your data sets so that they can even store it in their computing environment. So how do you handle all of this so that people don't end up having to play around a lot with, frankly, just writing different file paths and writing different, using different file formats? Redistribution is an issue that people can run into if you're working with, say, data that has personally identifiable information. You don't want to redistribute any data sets, right? So you really just want to make it so that somebody has access to the data set through whatever, let's say, commands that you ran to access that data set. And lastly, replication. You want some way of verifying that you're working with the same data set that the other person was working with when they ran their analysis pipeline. So since we're talking about reproducibility, and if you're familiar with version control software, you might say, why not just use Git for this, right? If you have something that's on the order of, say, like a couple of megabytes, that's a perfectly valid use of Git because it's very good at handling just plain text data. If you host your data set on GitHub so that other people can access it on the internet, 500 megabytes is okay. GitHub will complain about that. And Git is very good about handling changes in files, right? So if that data set isn't in fact static, but is potentially being updated over a couple of months, say, it's a great tool to just do version control on that data set as it's changing. But if you're working on, say, image classification tasks, and you want to run ImageNet, you can read through this little Wikipedia page, but essentially you'll find that you're not going to be able to readily process that or even store that on your computer. It's a huge data set of just images. So data depths, which stands for data dependencies, sets out to solve this problem by being able to handle any file within reason. It doesn't care about file size. Whether or not you're able to store that file on your computer is kind of your own issue, but at least data depth sets it up so that you know exactly what you're trying to download, and you know what exactly whether or not that's going to be doable on your computing environment when you try to access that data dependency. And this is a very important thing to note, is that this is really designed for static data sets. So if you want to update things, you're going to have to go in and do a little bit of work, but you can, this is really set up for a static workflow. Right, so I think from here we can just jump into really the magic of what data depths does. And this is, if you're a user, meaning that you just want to load some data set that somebody else used, this command here, which is a macro, data depth string, this is all you really need to know. So what this little incantation does is that when Julia evaluates this line here, what it's going to do is essentially try and track down where this data set lives and resolve it to a file path. If that data set doesn't live on your computer, it tries to download it from whatever source that the creator of that data set specified, or rather the creator of the data dependency registration block specified. So this gets around, any issue where somebody has to go in and figure out like, oh, I'm trying to run this software that somebody shared with me and wanted me to check. Oh, wait, I have to go back and download the data set, right? So this is all put into a very reproducible workflow, because all you have to do is call this one command to access it. Oops. So if you care about actually creating your own data dependency, you have to dig in a little bit more into the documentation, and from there you'll find this object called the data dep. This is just an object in the data deps package that allows you to specify this is what my data dependency is. So you give it some sort of name. You can attach a helpful message. This is a good place to, for example, acknowledge any papers that should be cited in association with that data set. List any license information that might be important for people to know. Or in some cases, you might have to specify some additional instructions in order to be able to handle that data. So again, a good place to put all that information. And then this third argument, which if you just want people to be able to download this file in a reproducible way, all you have to do is specify the location of that file. So if you're working in a lab that uses some sort of shared computing resource, that could mean that this remote path just specifies a particular location on, say, a different hard drive that you normally don't work with, right? So you can download it from there. If you're working with a file that's hosted on the internet, you would put in a URL here. Or if you, in one of my examples, you'll see you can actually use this to run a simulation pipeline to reproduce a particular data set that you then used in your own application, we'll say. So again, just to kind of recap here, if all you want to do is create this reproducible pipeline, you really need to care about the first three arguments. And optionally, it's very good practice. You might want to look at the fourth argument here, which is checksum. For our purposes, we won't talk about hashing functions or like how this verification process actually works. But essentially, the checksum is just a string that is used to verify whether or not the data set that you produced was, in fact, the original one that the author wanted to specify. Okay. So from here, there are a couple of additional options that you could specify, which is you can go in and actually configure how you fetch your data from the internet. So there's a custom argument that you can where you can pass in a function that basically overrides the default fetch method. So by default, when you actually try to use a data dep, what happens is the package goes in the background. It tries to determine whether or not that data dependency exists in your computer. If it doesn't, it will use, I think it defaults to something like base.download to retrieve that file from the internet or for whatever location you specified. So how do we actually use this in practice, right? We'll load up some of our packages here. You can read. Is this running? This is not running in the server, is it? Because the CSV should have loaded. Should I just reload the? Sure.
Sorry about that. Let me just. I just want to activate the environment up one level.
I see.
Oops. Join pass. Because I know the upper environment has the CSV.
Oh, right. It's the thing where it picks up. Yeah,
Yeah.
I should have put the
Sorry, I
project
should.
file in there. Yeah.
Down this up.
Okay. Okay, good. That loaded up, right? Right. We're really going to focus on the data deps package here, but you can just for your own reference, you can go ahead and look at the comments there. And this this code block here might seem a little strange. Essentially, I'm just setting it up so that we specify a particular file path where we're going to store all of our data sets that we download in this tutorial. Normally, you don't have to configure any of this. You can if you want to, but this is just strictly artificial for the purposes of the tutorial, because by default, data depths uses some heuristics to determine where to store your data sets if you don't configure it. Right, so we can get into our first example. This is using the famous iris data set in statistics. And all we're going to do is just download this file in a semi-reproducible way. So the first thing in creating a registration block is to just give a name for a data set, in this case, iris. And because I'm getting this from the UCLA machine learning repository, I have a little help string here that will display at download that just lets people know, like, this is where this file is coming from. Here's a very quick summary of the page on that repository to let you know what kind of data is in here. And finally, we're going to specify that URL, right, so that we know exactly where to retrieve the data. So we'll run this, and we just get a printout of the data depth block. And finally, we register this. So this is a key step. If you don't register your data block, data depths doesn't know what to do with it. So this is just saying, hey, please add this to the list of data dependencies that I've created, right? It's just keeping track. It's just bookkeeping. And finally, we get to actually using the data. And here we're going to call the data depth string macro, and we're going to give it the data dependency name. So this should just give us a prompt that tells us, we don't have this dependency on our computer, you should download it. So it gives us that help message that we wrote for ourselves. And finally, it gives you a prompt. Do you want to download this file from this location to this location on your computer? Right, and here you can actually confirm. We'll get a printout about the download. And finally, we'll get a warning message, because I had mentioned at some point that data depths uses some method of verification, right, to make sure that it's the original data set. Here it's just complaining because we never specified a checksum, so it doesn't know how to check that this is the original file. What you normally do is you actually provide a particular checksum at the registration step. So here it's telling us, here's the hash that I should have gotten if this is in fact the correct data set. So you can go back and do that. But for now, let's just check a couple things. So first thing to check is, did we actually download anything? Does this exist? Well, again, we're going to call the data depth string macro to obtain the file path. But in this case, I know that the file we downloaded is called iris.data. And so I'm going to use the CSV package to download it and pass it to the data frame object as a sink. And so if I run this, I'll get a printout of that, the first 5 rows of that data set. Right. Except in this case, there's a little bit of an error, right? And that I never specified whether there's a header, and CSV package is a little eager and detected a header where there wasn't one. So you can just easily fix this by telling it that there isn't a header there, and we get the correct readout. All right, so let's go back to that warning. We got this, here is just a quote of that message. The checksum is not provided. Add the data depth registration to the following hash line, with the following hash line. And so I created that same registration block from before, except I added the hash for the checksum step in this registration block. So I'm going to go back, register this data dependency, remove the file that we had just downloaded, and run the whole step again. So we're going to read in the first lines again, but again, because this is the first time, data depths will not find the file and will ask us to download it again, right? So we get that same prompt, and now we just confirm. And you can see this time we don't have that warning about the checksum. So as far as I know, this is the same data set that I was supposed to receive as intended by the author. So from here, there are a couple things that you can do. You can create all sorts of wrapper functions to immediately load your data in a particular format. You can incorporate this into a larger list of different data sets. For example, if you're developing some sort of package in Julia, you might want to make it easy for users to access a particular data sets that's used for a particular numerical experiment. Or you might want to do some sort of transformation of that data set that you downloaded to put it in a format that's suitable for your analysis or your application. So for that, we're going to look at another keyword argument, the postfetch method keyword. So this brings us to another example. For this, we're going to use some data on breast cytology. So you can look at this particular page on the UCI machine learning repository, and you can look at the data folder where the different files live. I'm just going to focus on this file here, breast cancer wisconsin.data and wdbc.data. They're two different data, closely related data sets. Okay. So for this, essentially what the postfetch method argument does is you pass in a function, and this function has to have this particular structure, which is it has to have a name. Well, it doesn't have to have a name, but you have to be able to pass in some sort of file path to it. In this case, it's going to be the location of the file after it's downloaded. So this runs after the download step in the data dependency pipeline. From that, you're free to do anything you want. So this can mean that you read in that data, apply some transformations, and possibly generate a new file that's saved on your computer. You can go in and modify the original file, whatever it is, but you're free to do whatever within this function. So to do that, I'm going to create another function that's not part of the postfetch method, but it is a step, which will create a standard format for our data set. In this case, I'd like because this is a classification data set, I'd like for the class labels to be in the first column and have the remaining columns in my table be features or variables or predictors. And from there, we'll be able to add another argument to allow us to specify, hey, this data set has a header, or this data set doesn't have a header, I need to create one. So this is going to standardize the table for us. You can look at the function itself on your own, but let's just check it against the iris data set here. Right, so I know that this doesn't have a header. I know that the class labels live in column 5, and I know that all the variables live in the first 4 columns. So once I do this, you can see that my data set has been transformed. Now I have the class labels in the first column and everything else in the subsequent columns, right? Nothing too crazy there. So we'll define a helper function that essentially allows us to apply this to the breast cancer Wisconsin data after we've downloaded it. So we want to save it in this particular format for our analysis. So the first thing we'll do is apply that standardizing function to it. So we're going to give it that local file path. In this case, again, there isn't a header. And I know for a fact that there are missing entries in this data set. So I'm going to specify the missing string. So it's going to, when we run our function on this, we're going to basically drop any records that include a question mark or empty strings. Finally, the class labels occur on column 11, and the variables occur in columns 2 through 10. And you can verify this by looking at the .names file inside that web page that I shared with all of you. So this is kind of the little table summary that's in there that helps you identify where the features live and where the class labels live. From there, I want to change the class labels because the original file has 2 for benign and 4 for malignant. I want to actually have the human readable stuff in there. So I'm going to specify benign and malignant for the class labels. And so that's what this block here does for us. And finally, we're going to drop anything with missing values. I don't want to include it for my analysis. The last bit here just goes through and defines a separate table that's not part of our data frame that just has the column label information, so that at some point someone can go back and retrieve, oh yeah, the first column corresponds to the diagnosis, column 5 corresponds to this particular variable or feature. And finally, I don't want to touch the original data set, so I'm going to save them as separate files that I give here. Okay, so we haven't done anything except define the function right now. We haven't actually applied it to our data. We're going to do the same thing for another data set that's included in that folder. We won't go through the detailed steps here. You can look at this on your own, but essentially we're running through the same thing. We want to standardize the format of the table. We want to change the class labels, and we want to do a little bit of processing on the pre-processing on the different columns that correspond to variables. So you can look at this on your own. But essentially, we have 2 files we want to download as part of this data dependency, and we have two separate pre-processing steps that we want to apply to each of those files individually. So how do we do this? Because I told you at the beginning, post-fetch method only takes one argument, right? It's one local file path. So the way that this resolves is that data depths will apply your post-fetch method in a sequential, I shouldn't say sequential, it'll apply that function to each individual file, possibly in a random order. But all we're going to do here is just add a check. If I have the breast cancer Wisconsin.data file, please apply this processing step. If I have the other file, wdc.data, apply this other function that I define. So that's it. That's all you have to do to stitch together the different steps. Okay. So now we'll create our registration block. This is essentially the same as before. We have our name. We have our help message. And in this case, we don't have a single path. We have multiple paths. So I just have the 2 URLs right there that you can check. And lastly, before our last thing, we have our checksum. And finally, we have our post-fetch method. So that's what we're going to do here, run that. And so let's try to load this data set now, or rather check the directory that contains our data sets. Nothing exists yet, so we're prompted to download, and we say yes. And you can see that it's downloaded each of those files individually, right? So let's check to see what actually lives inside the directory. We have, actually we're missing. Is that right? Oh, I see. Did we not run this? So if you're running all the cells in order, this should have worked. But I have a feeling that I skipped over a very critical step here. Yeah. I don't think I actually ran this. Let's try it again. Okay. So create the block and oops. And run this again, right? So the storage location changed. And I was using the wrong one before. So let's verify this went to, yeah, so we should have 4 files in here now. No, we only have 2. Let me see. Yep. Let's try running this and running this. This is the one I think I didn't run. Okay. It's fine. Okay, I'll troubleshoot this. Has anybody tried this and followed along and had it gotten working? You see 4 files there? Okay, I'm not crazy. So let's try to load in the one file that we do have here. You can see that our pre-processing steps were applied, right? We don't have the 2 4 labels anymore. We have benign and malignant. And we stripped the column information, or rather we standardize the column information so that everything reads as feature 1, feature 2 for our columns. And we also created this just small file to store the column names separately. You can see that we do in fact have diagnosis, lump thickness, and all that information there so that somebody can go back and retrieve that information. I'll try running this example, but I don't think. Yeah, I'll have to troubleshoot what happened here, but it seems like it's working on the actual server. Okay, so we'll move on to the third example here, which is creating a data set through a simulation and having that simulation be reproduced locally on somebody's machine. So in this case, I just have a function that generates a spiral pattern. You can read through the details if, oops. I see. We skipped over this. Let's abort. Okay. So let's run this here, right? So we have a function that generates a spiral pattern. I'll run this. This is using Maki to generate the little plot for us, so it might take a little bit. But in the meantime, I'll talk about the registration block for this. So this simulation is running locally on our machine. And we have a couple of different parameters that we can pass into that simulation function. The first argument here is a tuple that contains numbers for number of samples of each particular class. So there are 3 classes. In this case, I want 600 from class A, 300 from class B, 100 from class C. I want to pass in a particular random number generator with a particular seed and set a bunch of different simulation parameters as well, right? So in this case, by being forced to create this registration block, I'm forced to document exact settings I used to generate this data set, right? So it's not hidden in some supplementary data file that people lose access to over time. It's not documented in some obscure location. It's right there in the registration block, so anyone can look at this. If you're running my code to try and reproduce this, it's right there. And finally, we just take the simulation output and put it into a data file, right? So we can go up here. That's our spiral pattern. I actually generate a slightly different one when I run the simulation. But because I'm running this locally and not downloading a file, you can just pass in an empty string there for the download location, because what we're going to do is overload the fetch method instead. So we're not going to default to base.download. Instead, we're going to provide our own function that retrieves the data and generates the local file that we use later on in the data dependency pipeline. So we create our data dependency, we run data.string, and in this case, we get the same help message, but in this case, you'll see that unused is listed as the download location. So we're not really downloading this, but you'll see that we run our simulation, we store it into this directory, and then I can go back in and extract that file that we created through the simulation and obtain the particular pattern that I wanted users to use in their analysis. So down here, I have just a little aside that you can read on your own about random number generation, because this is something that I actually didn't know about. So just briefly, I'll mention that typically you're told, set a seed for your random number generator, and that it is sufficient to reproduce some of these results. But in fact, at least in Julia, I don't know how much development random number generators go through in other programming languages, but somebody might go in and change the random number generation algorithm for that particular version of language. So if you tried to use the default random number generator at Julia in the 0.7 days, and you try to recreate some or run some scripts, in version like 1.2 or 1.3, you might in fact end up with a different number string just because somebody made a slight modification to improve speed up in that random number generator algorithm, or fixed a bug that maybe doesn't affect your results. So maybe that level of reproducibility isn't crucial because your results shouldn't depend on like the statistical peculiarities of a particular random number generator. But if you really need that, there's a package called stable RNGs that you should use to make sure that you get the same random number string. But with that, just to recap here, do feel free to check out data.jl to help you document and reproduce your results so that other people can use them once you share it. And certainly use it if all you need to do is manage some local storage of data sets on your machine, because it makes the whole process pretty painless. Like you're not having to type in file paths into your different scripts. You just use that special macro to retrieve file paths based on data set name. So with that, I'll end it there.
In Python, there's something called, I can't remember the name honestly, but it is mentioned in the data depth documentation. I don't know that. Yeah, I just know the one that the data depth people mentioned in their documentation, but it's rare. It's something that's, my understanding, people have only started to pay attention to recently. I guess I forgot to share one little bit about this, but in some of my work, I was trying to use some data sets involved in cancer classification. So the labeled data that have a bunch of gene expression markers reported, right? And a bunch of labels for different types of cancers. Well, I was trying to find where those data sets were originally generated. And if I go through and find like the papers with that, created the microarrays that were used, you can find, links. And if you follow those links, you'll find that the file isn't posted. So I have no idea how to access those data sets anymore. And the funny thing is that there are papers from other people publishing in 2022 that are somehow accessing this. So I don't know how to compare my own results against what they're coming up with, because everyone is just kind of passing the files along to each other. So there's no way of knowing, if I can recreate anybody else's results, or if anybody can recreate my own results because of that. So it's an important area to pay attention to.
Thank you, Alfonso. Also, thank you for mentioning that stable RNG package. I myself encountered the issue. Just Julia version bumped, and then my testing example just didn't work anymore. Just because the random generator, they are not same across the different versions. It's very cool. It's a stable RNG.
Yeah, and it's something that people only started to pay attention to, at least in Julia.
Right.
It's like an offhand comment in the documentation, like, oh, by the way, it's not stable across versions. And now it's a lot more prominent. They make sure to mention it in the random number documentation.
Right.
Before, if you were using like Julia 1.0, no one would tell you that.
Yeah.
So
exactly.
You run into that issue.
Right. Okay. So is that we have 10 minutes or before the career panel?